My name is Sophia Magalhaes, and the title of my research is Region-Specific Maintenance of Blood-Brain Barrier Integrity by Astrocytes, Implications for Selective Vulnerability to Alzheimer's Disease. Alzheimer's disease, or AD, is characterized by hallmarks such as amyloid beta plaques, neurofibrillary tangles, and neuronal death. And what's interesting about these characteristics is they accumulate unevenly across the brain throughout AD, creating a pattern of selective vulnerability where certain regions of the brain are impacted faster and earlier in the disease than others. Two areas of the brain that have demonstrated distinct susceptibilities to AD are the prefrontal cortex, or PFC, which expresses vulnerability, and the visual cortex, or VC, which expresses resistance. The neuronal layers within both of these regions have additionally demonstrated differing rates of susceptibility to AD, with layers 3 and 5 being most vulnerable in the PFC and layer 4 being most vulnerable in the VC. Now, studies like Lang et al. In 2021 have characterized these selectively vulnerable neuronal populations, while Groth et al. In 2018 found that regional vulnerability to AD is related to molecular properties. But limited research has been conducted on the contributions of neuronal environments to selective regional and layer-specific vulnerability to AD. One of the most prominent cell types in these neuronal environments are astrocytes, which have been shown to support neurons in numerous ways. One of their most important functions is maintaining the integrity of the blood-brain barrier, or BBB, which they do by unsheathing blood vessels with their processes. But if these processes begin to degrade, this compromises the integrity of the blood-brain barrier, which may lead to neurodegeneration noted in areas of specific vulnerability to AD. However, the relationship between regional vulnerability to AD and impaired BBB integrity is not well understood. This leads to the purpose of my study, which was to determine if astrocytic failure to maintain BBB integrity corresponds with regional vulnerability to AD, as well as to determine if layer-specific vulnerability is promoted by different levels of astrocytic coverage of blood vessels across AD. I hypothesized that blood vessels in the PFC would experience reduced astrocytic coverage as the severity of AD increased, while blood vessels in the VC would maintain stable levels of astrocytic coverage even as AD became severe. So this was done by measuring 12 humans pre-mortem according to their clinical dementia rating, or CDR, which was done by an outside collaborator. Humans were placed into either a control group of CDR 0, a mild dementia group of CDR 0.5, or a severe dementia group of CDR 3. Post-mortem, brain tissue samples were utilized and analyzed to evaluate Thal stage, where humans are placed into either a control group of stage 0, a pre-AD group of stages 1 through 3, or an AD group of stage 4. And additionally evaluated based on Brock stage, where humans were placed into either a control group of stages 1 and 2, or an AD group of stages 3 through 6. Now, multiplex immunofluorescence staining was conducted on Brodmann areas 9, 10, and 46 of the PFC, and Brodmann area 17 of the VC, also known as the V1, and markers of interest for my study were ALDH1L1, which labeled astrocytes, GFAP, which labeled reactive astrocytes, and collagen 4, which labeled blood vessels. Additionally, all tissue was counterstained using DAPI, which binds to DNA, and from this we were able to layer images in order to create quality control scores that labeled areas of the tissue that were damaged or altered throughout the staining process. I conducted all image analysis independently in the QPath software and wrote code to automate cellular detections and calculations. Now this is an example of an image that I will look at. And if we zoom in here, you can see my exact areas of focus were overlap between astrocytic markers and vasculature annotations. So I calculated astrocytic coverage by dividing astrocytic area by vasculature area and multiplying that by 100 to get a percentage. And I normalized this data per image by dividing that value by the percentage of astrocytic coverage respective to each image. All data was exported and analyzed using RStudio, and I first looked at astrocytic coverage across CDR levels regionally and found that GFAP-specific astrocytic coverage of blood vessels maintained relatively stable as AD severity increased in the V1. Looking at layer-specific coverage of collagen 4 across CDR levels, I found that there was a significant increase in GFAP-specific astrocytic coverage of blood vessels in layer 4 of the V1, which is interesting to note as this is the most vulnerable layer in this region. Next, I looked at astrocytic coverage across Thal stages regionally, and again found that GFAP-specific coverage of blood vessels maintained relatively stable in the V1 as AD severity increased. Looking at layer-specific coverage of collagen 4 across Thal stages, I again found a significant increase in GFAP-specific coverage of blood vessels across AD severity in layer 4 of the V1. Lastly, I examined astrocytic coverage across Brock stages regionally and found that GFAP-specific coverage maintained relatively stable as AD severity increased in the V1. But looking at layer-specific coverage of collagen 4 across Brock stages, it can be noted that GFAP-specific astrocytic coverage of blood vessels significantly increased as AD severity increased in layers 4 and 5 of the V1. Now, I found that astrocytic coverage did not significantly change across CDR, Thal stages, or Brock stages regionally, but this indicates that in the V1, astrocytic coverage maintained relatively stable as AD severity increased, meaning that astrocytes in this area did not degrade in response to the severity of the disease. I additionally found patterns of increased astrocytic coverage as AD became more severe, specifically when reactive markers were expressed, which may be explained by Lawrence et al. In 2023 and Lee et al. In 2022, which concluded that reactive astrocytes may undergo alterations in shape and function that contribute to a neuroprotective phenotype. And this may mean that astrocytes in this layer, being more vulnerable, are adopting a more neuroprotective function by increasing their coverage of blood vessels, providing more support to their environment, and contributing to the region's overall resistance to AD. Now, my study provided a greater understanding of the role that astrocytes play in selective vulnerability to AD, as well as an indication of region-specific astrocyte reactions in populations, since that increased astrocytic coverage was only noted in layer 4 of a resistant region. And this implicates biomarkers of resistance and targets for future AD treatments that may aim to promote brain-wide resistance to AD. Now, my study utilized a relatively small cohort size, so future studies should utilize larger cohort sizes to validate these results. Additionally, tissues did face certain multiplex-related alterations, but quality control scores really limited the impact of this limitation on my study, as any altered areas were not considered in data collection. Lastly, I detected a lot of variation in the fluorescence intensity of astrocytic markers, so future research should aim to develop more precise methods of annotating astrocytes, which is something I'm currently working on with my lab. In conclusion, my hypothesis that blood vessels in the PFC would experience reduced astrocytic coverage as the severity of AD increased was not supported, but I found that blood vessels in the VC maintained stable levels of astrocytic coverage even as AD becomes severe. I also found that astrocytic coverage significantly increases in neuronal layers of vulnerability in the V1, especially when markers of reactivity were expressed, suggesting that the presence of reactive astrocytes may contribute to neuroprotection and calling for a greater need to understand the impact of reactive astrocytes on regional vulnerability to AD. I would like to thank my mentors for their guidance, my teachers for their support, my family and friends for their love and encouragement, and you for listening. Thank you so much.